No.
Okay.
No, apoptosis?
No, the piece that T cell.
Oh, epitope.
That's one of the pieces that it's presenting, right?
Yeah, an epitope just refers to the part of the antigen that actually binds to the surface receptor. Okay, so this is the part we talk about when you make vaccines and things like that from a toxin or a bacterium. As long as you kept the antigenic epitope, what we're meaning then, as long as you kept, say this is a full bacterium, as long as you kept the part that bound to the receptor, the surface receptor, that's the epitope, you'll still get the immune response. And so that's why we can do vaccines with bacteria and stuff. If we kill the microbe and keep that piece of protein that causes the immune response, that's all we need to expose your immune system to. You know, just the epitope that will bind to this particular receptor. So a lot of times you talk about a bacterial cell, you know, as the antigen, but this bacterial cell can have several different epitopes on it that are recognized by different B cells. Okay. So natural killer cells, we pretty much covered them. They're going to recognize antibodies, so they're part of the ADCC, antibody-dependent cellular cytotoxicity, and they're going to recognize cells that are lacking MHC molecule. Okay, so if you don't have an MHC molecule, meaning something's wrong. If that cell is lacking that, they can't present internal molecules for checking, and so maybe they're already infected. There are some bacteria and viruses and stuff that can cause cells to pull in their MHC molecules or lose their MHC molecules as they change the surface of the cell by studying it with their own molecules or things like that. So natural killer cells will not only kill cells that are bound by antibodies, but they'll kill cells that are lacking MHC molecules as well. Which brings us to selection of T cells and B cells. Remember that they are produced in the bone marrow. All white blood cells are produced in the bone marrow. And the B cells mature there. And they're in the bone marrow. They undergo negative selection. And this process here has the B cells being exposed to self molecules, self antigens. If the B cell recognizes any of those self molecules, then it's going to be destroyed. It's going to be selected against. If it doesn't recognize anything, then it will go on and mature and then be released into the lymphatic system eventually in hopes of finding the antigen of its dreams. T cells, on the other hand, they're going to undergo two types of selection. First, they're going to undergo positive selection in the bone marrow. The T cells?
T cells undergo positive selection?
They undergo two selections. They undergo a positive selection in the bone marrow. Here, they're exposed to MHC molecules, or cells with MHC molecules, and if they recognize MHC molecules, they're selected for. We want our T cells to be able to recognize MHC molecules. If they do, then they'll make their way to the thymus where they'll mature. There in the thymus gland, they'll be exposed to self molecules now. And negative selection will take place then in the thymus. Go ahead.
So they have to be
exposed to the MHC molecule to make their way to the thymus?
Right, so in the bone marrow, because what we want to do, the T cells have a tougher job than the B cells, okay? We just want to make sure that the B cells don't recognize our own molecules, okay? So we just expose them to our own molecules, and if they don't, they're good to go, and we release them. If they do, we destroy them. But the T cells, they have to undergo dual selection. They have to undergo a positive selection in the bone marrow. They're exposed to cells that have MHC molecules, and if they recognize those MHC molecules, they're selected for. Okay? From there, they'll make their way to the thymus gland where they undergo negative selection, exposed to self molecules, and if they recognize self molecules, that's bad, and they'll be destroyed.
And at that point, they haven't differentiated into T helper to cytotoxic?
No, because they haven't been presented anything from the dendritic cell yet. They're still in the thymus. They haven't made it into the lymphatic system, so no, they're still naive, if you will. One more thing that we want to say about this chapter before we move on, the antibody. Understand the makeup of the antibody, because it'll be very important in some of the questions I've asked you. Remember that generally a Y-shaped molecule, and the active regions consist of the claw and the stem, if you will. This area up here, also known as the FAB region, is the area that will bind the antigen. And this area down here, known as the FC region, is what the macrophage will bind to. That way we only have to have the macrophage recognize one or a few different molecules. They don't have to recognize 100 million different molecules. Yes?
The FAB region is the left arm have a different sequence of their clones of each other.
Right.
Like that part I wasn't sure.
No, they'll be identical binding sites on both sides. Chapter 18, chapter 17 dealt with post-microbe interactions. This really, I think, was a chapter more about terminology than anything. I mean, there's a lot of terminology in here. Most of it you're probably familiar with, but I would recommend that you be sure you understand what it is. Again, we talked about normal microbiota in this chapter, so I won't go over that again. We talked about how infections take place, colonization, pathogenicity, virulence, communicable disease, contagion, infectious dose, all terms that you need to be familiar with. Opportunistic infector, primary infector. So, one thing to keep in mind in infections, we can label them as being primary infections. These are infections that will cause a healthy individual to get sick. And then we can also identify secondary infections, which come to a person that's already got a depressed immune system for whatever reason, probably because they're fighting off another infection. These guys are opportunistic, and opportunistic infectors often are part of your own normal microbiota. Recognize that the infectious dose has a lot to do with the microbe's ability to avoid your host defenses. So we've used this example before. Vibrio cholera has to make it through the acidity of the stomach before it can get to the small intestine. And that kills a lot of the cells. So you really have to ingest hundreds of thousands of Vibrio cholera cells in order to get sick. However, Staphylococcus aureus has many different mechanisms for avoiding our immune system. So as little as 30 cells can cause an infection. So each microbe, depending on their defense mechanisms, their ability to get around our defense mechanisms will dictate the infectious dose. Virulence. Again, we're talking about a degree of pathogenicity, if you will. Now, how many individuals will get sick and how violent will they get sick? You know, how severe will those symptoms be? Mentioned the course of the disease, you know, the incubation period, the illness, the convalescence. We talked about duration, acute, chronic, latent, distribution of bacteria are localized versus systemic or generalized. And then we talked about two postulates, Koch's postulates and the molecular postulates. Understand that these postulates were put in place so that we had a mechanism for identifying which microbes cause disease. You might be sick, but you probably have several different microbes that are inhabiting your body. And we need to identify which one of them is responsible for the disease. So Koch's postulates isolated the microbe from the sick individual, grew it in pure culture, exposed someone to it, made them sick, and then isolated it from them to prove that that was the microbe. The problem with that is you can't expose everybody to disease to see if it'll make them sick. So the molecular postulate kind of did away with it. With the advance of genetics now, all we have to do is knock out the virulent gene, the gene that produces the product that causes the illness. If you knock out that gene, then you prevent the microbe from spreading, but it also helps us identify. So get back to this molecular postulate here. Identify the virulence factor, the gene that's responsible for it, knock it out, and the microbe shouldn't make anyone sick. Restore it, and its virulence should come back. Establishment of an infection. Recognize you've got to have adherence. You know that the microbe has to adhere to the cells. And so it means the cells has got to have the appropriate receptors in order for adherence to occur. Colonization's got to take place. The microbes have got to grow. And then, in many cases, there has to be a delivery of the effector molecule. Now, some cells, as we talked about with the mechanisms of pathogenicity, that some microbes, you know, all they do is just grow. Their presence is the fact that they're multiplying on mucous membranes. But many of them have something that they deliver, you know, a molecule that causes the cell to take them up, or, you know, a molecule that causes the cell to release its electrolyte, or something like that. And so, for the infection to be complete, the delivery of the effector molecule has to take place. So a lot of times, if you can block that, you can slow the infection. We talked about several different methods that the microbes use to avoid host defenses. So be sure that you're familiar with those different mechanisms. Antigenic variation, something I've thrown up a couple times. Be sure you understand what that is. We talked about several toxins too, AD toxins, membrane damaging toxins, and super antigens. Basically, know that those are exotoxins. Those are toxins that are released by the cell, and they differ from the endotoxin. Endotoxin is actually part of the gram-negative cell, part of that outer cell wall. That brings us to chapter 18, the hypersensitivities, 4 of them. Okay, So you should know each of the hypersensitivities and a little bit of detail about them. Because this lends itself to a number of different questions. You know, a multiple choice, or excuse me, a true-false question. Type 1 hypersensitivity involves a T cell response. Okay, but then I can use a multiple choice, where I could say the same thing and then give you, 4 options. And then I have 4 hypersensitivities to choose from. So I really have 16 different questions here that I can ask you with regards to these hypersensitivities. So be sure that you know each of the hypersensitivities and what they do or what they are. So type 1 was the immediate IgE-mediated hypersensitivity. This involved, that first exposure to the antigen, a sensitizing of mast cells, and then second exposure causing a major release of histamines, and then the allergic response that you observe. Type 2 was the cytotoxic hypersensitivity. This dealt with the blood types. And here we talked about the RH blood type specifically. So be sure you understand